Have a good day. I am Dr. T. Elakkiya, Assistant Professor, PG and Research Department of Chemistry, Sri Sharda College for Women, Autonomous, Salem 16. Today, we will discuss about NMR spectroscopy, proton NMR, N15, F19 and P31. Overview of NMR spectroscopy, then applications of proton NMR spectroscopy, applications of N15 NMR spectroscopy and applications of F19 NMR spectroscopy, applications of P31 NMR spectroscopy. Overview of NMR spectroscopy. NMR is based on the interaction of nuclei with the spin in an external magnetic field. Nuclei absorb radio frequency energy at a specific resonance frequency. The chemical shift indicates the electronic environment around the nucleus. Spin-spin coupling causes splitting of signals into multiplets. NMR spectra help to determine molecular structure and identify compounds. Principles of NMR, basics of nuclear magnetic resonance. NMR is a powerful spectroscopy technique that exploits the magnetic properties of atomic nuclei in magnetic field to deduce information about molecular structure. Key terms and concepts: nuclear spin, chemical shift, resonance, spin-spin coupling, relaxation time. That means these are the essential terminology in NMR studies. Nuclear spin, property of certain nuclei that makes them NMR active. Chemical shift, position of NMR signals indicating the nucleus environment. Resonance, absorption of RF energy by nuclei in a magnetic field. Spin-spin coupling, interaction between neighboring spins causing signal splitting. Relaxation time, that is T1 by T2, time taken for nuclei to return to equilibrium after excitation. So the essential terminology in NMR includes chemical shift, coupling constants and resonance frequency, each crucial for interpreting spectral data and understanding molecular environments. Instrumentations in NMR. Components of an NMR spectrometer. So, an NMR spectrometer comprises several key components including the magnet, RF transmitter, sample probe, receiver, detector, computer system, etc. Magnet, it provides a strong and stable external magnetic field. RF transmitter generates and sends radio frequency pulses to exit nuclei. Sample probe holds a sample tube and detects the NMR signals. Receiver or detector, it detects emitted RF signals as nuclei lags. Computer system processes signals to produce the NMR spectrum. Proton NMR. Sample preparation techniques. Proper sample preparation is vital for accurate NMR results, involving the selection of suitable solvents, concentration optimization, and ensuring sample purity to avoid interference in spectra. So, applications of proton NMR spectroscopy. It is mainly used for organic compound analysis. Proton NMR involves determining the molecular structure of organic compounds by analyzing chemical shift, coupling patterns and integration of signals. Mainly, chemical shift shows the types of hydrogen environments. Example, aromatic, alkyl, aldehyde. Integration indicates the number of protons contributing to each signal. Multiplicity, that is splitting, reveals how many neighboring protons are present. Coupling constants give information about bond angles and distances. Combined, these features help to deduce the molecular structure of organic compounds. Studying chemically complex mixtures with a proton NMR. Proton NMR is instrumental in analyzing chemically complex mixtures, allowing for the identification and quantification of individual components in mixtures such as essential oils or food products. Yes. It identifies multiple components by their distinct chemical shifts. Its integration helps estimate the relative concentration of each compound. Peak overlap is common, requiring techniques like two-dimensional NMR for clarification. Solvent suppression is used to reduce interference from common solvents like water. It enables non-destructive, rapid analysis of chemical mixtures like biological fluids or extracts. It is mainly used for biological applications. Proton NMR in metabolomics studies. Proton NMR is widely used to profile metabolites in biological samples. Example, urine, serum. It provides quantitative and qualitative data without extensive sample preparation. That is very important. Chemical sieves help to identify metabolites based on known reference spectra. Multivariate analysis. It is used to interpret complex NMR data. It aids in biomarker discovery, disease diagnosis, and metabolic pathway analysis. So it enables the profiling of metabolites in biological samples, assisting in biomarker discovery and understanding metabolic pathways in diseases. Proton NMR in drug discovery and development. Proton NMR plays a critical role in drug discovery by investigating molecular interactions, compound characterization, and providing insights into the pharmacokinetics and behavior of potential drugs. It identifies lead compounds by analyzing their structure and purity. Helps to study drug-target interactions through chemical shift changes. It supports metabolite profiling in pharmacokinetics and metabolism studies. It assists in formulation analysis and stability testing of drug candidates. It enables non-destructive, reproducible analysis throughout the drug development pipeline. Applications of N15 NMR spectroscopy. Protein structure determination. N15 labeled proteins are used to study backbone amide groups in NMR. Heteronuclear single quantum coherence spectra map each NH pair, providing a protein fingerprint. It enables assignment of residues and monitoring of structural changes. Multidimensional NMR, example 3-dimensional, 4-dimensional, helps determine full 3D structure of proteins. Useful in studying protein folding, dynamics, and ligand interactions in solutions. So isotope labeling techniques involve incorporating N15 into amino acids of proteins, allowing detailed examination of their 3D structures via NMR studies. Interaction studies using N15 NMR. N15 NMR is used to monitor changes in chemical shifts during protein-ligand or protein-protein interactions. Binding events cause altered chemical environments of involved residues visible in the spectra. Chemical shift mapping reveals specific binding sites and interaction interfaces. The technique provides insights into kinetics and thermodynamics of interactions. It is valuable for studying dynamic interactions in solution without recurring crystallization. Interaction studies using N15 NMR can reveal insights into protein-protein and protein-ligand interactions, providing information on binding affinities and dynamics. Nucleic acid research. N15 NMR in analyzing RNA structure. N15 labeled RNA allows monitoring of nitrogen atoms in the RNA backbones and bases. NMR spectra help design resonance to specific nucleotides, revealing their environment. Two-dimensional and three-dimensional NMR techniques like MOESPI assist in determining spatial proximities of nucleotides. Conformational changes in RNA, secondary and tertiary structures can be studied upon ligand binding. It enables the exploration of RNA folding, dynamics, and interactions in solution. Hence, N15 NMR is essential for analyzing RNA structures, enabling the study of base pairing, folding, and dynamics under various conditions. N15 NMR in hybridization experiments. N15 labeled nucleic acids are used to study hybridization events between complementary strands. Chemical shift changes upon hybridization reveal the interaction between strands. NMR provides detailed information about sequence-specific binding and duplex formation. Multidimensional NMR helps track the kinetics and thermodynamics of hybridization. It offers insights into the stability and conformational changes during hybridization process. Hybridization experiments with N15 NMR labeled nucleotides enhance the understanding of the binding mechanism and stability of nucleic acid complexes. Applications of F19 NMR spectroscopy. Fluorinated compounds. Tracking fluorine in metabolic pathways using F19 NMR spectroscopy allows researchers to monitor the distribution and metabolism of fluorinated compounds in biological systems, providing insights into metabolic activity and pathways. Accessing molecular interactions, F19 NMR can analyze interactions between fluorinated compounds and biomolecules, offering critical information on binding affinities and structural changes in various biological environments. It is mainly used for industrial applications, particularly pharmaceutical applications. In the pharmaceutical industry, F19 NMR plays a key role in drug design and development by allowing for tracking of fluorinated drugs, optimizing their efficacy and safety profiles. Material science insights. F19 NMR spectroscopy is utilized in material science to investigate the properties of fluorinated materials, enhancing the understanding of their performance in various applications, including coatings and polymers. Applications of P31 NMR spectroscopy. It is mainly used for phospholipid studies, particularly membrane composition analysis. P31 NMR spectroscopy is utilized to identify and quantify various phospholipids in biological membranes, providing insights into cell membrane architecture. Integration with other techniques. P31 NMR in studying liposome formulations, which are essential for drug delivery systems, examining size, encapsulation efficiency and stability. It is also used for environmental monitoring, soil and plant studies, that means water quality analysis. So P31 NMR spectroscopy in accessing phosphorus forms in soil and studying plant uptake processes, contributing to soil fertility and agricultural practices. P31 NMR for analyzing phosphorus species in water bodies, aiding in the monitoring of eutrophication and overall water quality assessment. Thank you.